Okay, let's start. Great. So good afternoon, everyone. So this is the our DFCI oncology seminar series. Is my voice okay? Yeah, okay, good. So it is really my great pleasure to introduce our speaker today, Dr. Ben Neill, a man who needs no introduction. But just let me do a very brief introduction just so that, complete my assignment. So Ben is professor at the Department of Medicine, NYU, and the director of Palmer Cancer Center. So where Ben is leading both a team of scientists working at the forefront of preclinical research and also a group of physicians in the clinic to provide cancer patients with novel therapeutics and treatments. Ben received his Bachelor of Art from Cornell in 1977, PhD from Rockefeller 882, MD from Cornell University Medical School 883. And then he was New York, and then he came to Boston from 883 to 88. 1983 to 1988, Ben was resident at the Beth Israel Hospital, and then also same time he's a special fellow and a postdoctoral fellow at Harvard University here in Cambridge. He was recruited as the assistant professor of medicine at Harvard Medical School, and he became associate professor of medicine and then full professor in 1999. And he was named William B. Castle Professor of Medicine in 2006. Then he moved to Toronto. So he was there in 2007 to 2015. And then he was professor there and at the University of Toronto in Canada. And then he was director for research at Princess Margaret Hospital Cancer Center. And he was also a founding director of the Campbell Family Institute for Cancer Research. And then so good. I think that was the big news or the big deal that NYU Medical School recruited him back to New York. And since then, he is professor of medicine there. And then he's also the director of Laura Isaac Palmer Cancer Center at NYU Medical School. So of course, he received numerous honors and awards in his early years, in his career in 1980s and 1990s. He was already a rising star. He received many awards, including special fellowship from Leukemia Society of America, Harvard University nominee for Pew Scholars Program, American Cancer Society Junior Faculty Research Award, AACR, Gertrude Elim Award. I think it was like a really famous award. And then more recently, Ben has been elected AACR Board of Directors, elected member Association of American Physicians. So Ben's research focuses on cell signaling in cancer and disease, as well as the biology of breast and ovarian cancer. And he has authored more than, I think almost 300 original research papers, more than 30 reviewers, and a number of books. So today, Ben's seminar title is New Approaches to Target Cancer. So Ben, the floor is yours.
Okay.
Take it over.
Okay.
Oh, forgot I said anything. So please, audience, post your question on the Q&A tab, not the chat tab, Q&A tab. Okay. Okay, Ben, now yours.
Okay, well, thanks, Jean. That very, very generous and very long introduction, which only makes me feel very old. So
I'm sorry.
Thanks for inviting me. It's always a pleasure to get this far, but I want to apologize in advance. I had a choice between being home with the dog and having the helicopters from the hedge fund managers who increasingly leave on Thursday afternoon to go to the Hamptons instead of on Friday. So there may be some noise in the background, and I apologize. I can't really do much about that. These are my disclosures. So the three that are relevant are shown in red. Navir Pharmaceuticals which I'm a co-founder, makes SHP2 inhibitors, although I won't be talking about those drugs today. My wife owns shares in Amgen, which makes G12C inhibitors, which I will be talking about today, although not their inhibitor. And I've also been an expert witness in federal litigation on talc and ovarian cancer. I'm going to be briefly talking about ovarian cancer today, but nothing having to do with talc. So if I put on my cancer center hat, cancer center director's hat, and I think about where we are in the field today in cancer medicine, I think it's true that conventional modalities such as chemotherapy and radiation are still the mainstay. And it's important to remember that these do cure several malignancies. We also have targeted therapies, and maybe that's a little bit of a misnomer. I prefer to think of them as signal transduction therapies. And we've learned a lot about these therapies. We know that resistance always develops to signal agents with these drugs, and that combinations of these agents are generally toxic, or if they're not, resistance develops to the combinations too. And I think it's fair to say that, for real cancers, targeted therapies extend life and they don't cure. And I prefer to think of targeted therapies as sophisticated debulking. Now, we know that immunotherapies such as checkpoint blockade and CAR T cells can cure, but most tumors don't respond. And I've increasingly come to believe something that I'm sure most of the immunologists at the Farber already believe, which is that all cures of cancer are immunologic, either accidental, as induced by conventional modalities, or intentional, like with checkpoint blockade and CAR T cells, and that the only way to cure cancer is to generate a durable anti-tumor T cell response. Now, if you look at cancer biology in 2021, we know that targeted therapies are generally tested against standard cell line-derived xenografts, patient-derived xenografts, and more recently organoids. And all of these systems lack an immune system or have an incomplete immune system. Immune therapies, by contrast, are generally tested against a few syngeneic models, or so-called syngeneic models, or occasionally against genetically engineered mouse models. For the syngeneic models, the cell of origin of the tumor, which is critically important to how it's going to respond, and or the genomics are wrong, limited, or unknown. And there's also problems with GEMs in that you generally don't have a lot of combinations of cancers for a given GEM. And even when you do, most of them are designed so that you have field effects, and that's not really the way cancer develops in vivo. So I think that what we need for progress is to focus on cures. That's stealing a word from a phrase that I first heard from Bill Sellers at a meeting in Italy when I was in Toronto. Not cancer as a chronic disease, cures. And in order to do that, I think we have to consider all drug effects on tumor immunity, understand the ontogeny and the vulnerabilities of resistant cells in response to initial therapies. We need better models with defined and relevant tumor genetics. And ultimately, we're going to need probably some new agents, but even presently, rational new combinations that may be combinations of conventional targeted and IO drugs. So what I'm going to do today is cover three aspects of what I think we need for moving forward in cancer. One I'll just briefly mention at the beginning, because I'm not going to present any new data on that. But first, I'd like to talk about developing SHP2 inhibitor combinations for targeting the RAS-ERK pathway. And this stems from several papers, including one that's not actually in press, it's not actually in print yet, it's online. Second, understanding the ontogeny and vulnerabilities of resistant cells, and this is a preprint that I'll talk about, and we're in revision for cancer discovery. And then just briefly mention this, at the beginning of the talk. So, okay. So I just wanted to call attention to what's becoming a major emphasis in our group that basically tries to integrate several of the features that I illustrated on the previous slide, which is to develop new models that are immunologically competent, genetically defined, and can be used to iteratively test combination therapy. So this is a work that was published earlier. Actually, the print version was earlier this year. The online version was last year by Shuang Zhang, a former postdoc in the lab. And it stemmed from her development of a fallopian tube organoid, epithelial organoid system that reproduces the differentiation properties of normal fallopian tube epithelial cells. And these cells can be engineered by either lentiviral or retroviral overexpression and or CRISPR-Cas9 editing to develop defined models that represent the major subtypes of human ovarian cancer. And then these cells can be cultured indefinitely in vitro in three-dimensional cultures or in two-dimensional cultures and subjected to a wide variety of assays. And then based on the results from those initial studies, one can test various agents in tumors by reimplanting these syngeneic organoids into the orthotopic site, the bursa of the ovarian bursa. And then we can assess the efficacy of drugs alone and in combination and iteratively try to develop new therapies. And I'm not going to show you any data on this. We've also developed human ovarian organoids, fallopian tube organoid systems, and have organoids from human patients with ovarian cancer. And so we can put all of these together and try to develop rational combinations. And we were able to develop a curative combination to a highly aggressive cyclin E tumor. And it's in this paper if you want to read about that. And we're doing a lot more studies on this, but they're not complete yet or ready to talk about. So what I want to spend most of my time on are the other two stories, which are further towards completion. The first is this recently published story that has to do with a longstanding interest of our lab, SHP2, which is encoded by the PTPN11 gene, so you often won't hear it called PTPN11. So this is the cartoon-like structure of SHP2 or PTPN11, which our lab identified in 1992 when I was in Boston early on. It has two N-terminal SH2 domains, a central catalytic domain, and a C-terminus with two tyrosines that play a modulatory, although not a central role in RAS-ERK activation in most cells. It's expressed ubiquitously, and that makes it very important to understand the effects of SHP2 inhibitors, not just on the tumor cells, but on cells in the microenvironment, because SHP2 plays a role in a large number of signal transduction pathways. It regulates cell survival and proliferation primarily through activation of the RAS-ERK signaling pathway. Exactly how it activates RAS still remains controversial, although I think there's general agreement now that it acts upstream of the exchange factors SOS1 and SOS2. Germline activating mutations in SHP2 cause a genetic disorder called Noonan syndrome, and somatic mutations are associated with rare forms of leukemia in childhood, or myeloproliferative disease is more accurate in childhood, and also several solid tumors. It's also a potential effector of some immune checkpoint receptors, although recent data suggests that it's not essential for PD-1 action. It is definitely associated with PD-1. And the reason why it's a topic of great interest now is because multiple groups, including the Vir, which I mentioned, but several others, have SHP2 inhibitors in the clinic. So these inhibitors basically exploit the SHP2 regulatory machinery that was first worked out from biochemical studies that we did in collaboration with Chris Walsh's lab in Boston, and really were elegantly worked out by the initial crystal structures on SHP2 that were carried out by Mike Eck and Steve Scholson across the street. Sorry. So what these structures showed was, I hate Zoom. How do I go back? Okay, so in the basal state, and by the basal state, I mean in the absence of a phosphotyrosine peptide that binds the N-terminal SH2 domain, the NSH2 domain is inserted into the catalytic domain, basically blocking the active site. Now, the phosphatase domain also contorts the NSH2 domain, so there's mutual allosteric inhibition between the NSH2 and the PTP domain. And so most of the ligands for SHP2 are bisphosphotyrosine proteins, and presumably the CSH2, which is available, will bind first, and then that juxtaposition of a high local concentration of the other phosphotyrosine can overcome this mutual allosteric inhibition and lead to activation of the protein. So this elegant molecular switch functions to ensure that SHP2 is only activated in the right place in the cell at the right time. So workers originally at Novartis Pharmaceuticals, based on an elegant screen, realized that there was an additional pocket on the backside of SHP2 that's formed by the junction of the NSH2, CSH2, and PTP domains in the closed form, and that if one put appropriate chemical matter into this pocket, you could stabilize the pocket and prevent the kind of loop openings that are necessary for this allosteric switch to turn on. And I'm going to be talking today about a tool compound, SHP-099, But the SHP2 inhibitors that are in the clinic all basically bind the same pocket and exploit the same regulatory mechanism. So because that pocket is formed only in the closed form, most activated mutants, certainly all the strongly activated mutants that are associated with cancer as oncogenes, are inhibitor insensitive. So the reason, however, why SHP2 inhibitors are potentially valuable for cancer is because they target a lot of other situations that occur in the disease, such as RTK amplification and gratuitous activation of downstream signaling pathways by, for example, PTK fusion proteins. But there's a more broad set of actions that SHP2 inhibitors might work on in adjunct with other signal transduction inhibitors. So in 2018, several labs, including our own, found that SHP2 inhibitors could abrogate adaptive resistance to MEK inhibition. And so for those of you who aren't familiar with this phenomenon, when you inhibit MEK, and therefore the downstream ERK pathway, that leads to de-repression of a number of genes for growth factors and their cognate RTKs. And then these growth factors can signal back through, initially we thought only wild type SHP2, but it's also clear, and I'll get to in a minute, that it can block activation of certain so-called cycling mutants of RAS. And then that can overcome the action of the downstream signaling inhibitor and result in resistance to that drug. So when you block with SHP-099, you can prevent this feedback loop and get increased efficacy of MEK inhibitors at tolerable doses of both agents. Now, as I mentioned, RAS mutants are not all the same. And it was appreciated about 10 years ago now that certain RAS mutants, although all of them are relatively refractory to GAPs, certain RAS mutants, most notably G12C, but a few others, maintain sufficient intrinsic GTPase activity that if you block upstream activation, they will discharge their GTP to the GDP state and become inactive. And so the RAS inhibitors, the covalent RAS inhibitors of G12C capitalize on this by finding a pocket that's present only in the RAS G12C GDP state. And so in a way, you can imagine these inhibitors are competing with SOS1 and SOS2, the exchange factors, because if SOS1 is highly active, it'll immediately recharge to the GTP state where these inhibitors can't bind. So again, our group and several others reasoned that SHP2 inhibitors, by blocking the action of SOS1 and SOS2, could increase the occupancy available of the GDP state and therefore the activity of compounds such as this tool compound RH1620 and also the now approved Amgen 510 or sotorasib and other RAS G12C inhibitors. And we also identified in the study that we did some very interesting effects on the tumor microenvironment, and it was those effects that we've now followed up in lung cancer. So the questions that I'm going to address in the first part of the talk is, what about other somewhat cycling RAS mutants like G12D, which does have some ability to cycle, but it's nowhere near as cycling as the G12C mutation. The other thing was that we noticed that none of these combinations was curative, and we wanted to know why that was. And so we wondered whether SHP2 inhibition, in addition to potentially beneficial effects on the tumor microenvironment, might also have some deleterious effects, and if we could intercept or interrupt those deleterious effects and increase the efficacy of SHP2 inhibitors. So the initial work that we did in this setting, we did in collaboration, well, all of the work on this part of the talk, we did in collaboration with my colleague Kwok Wang, and used initially the G12D P53 knockout lung cancer model. And this is really kind of a metastasis model because we're injecting these KP cells into the tail vein. They then form multiple metastatic deposits in the lung that can give you a very large tumor load and can be followed by serial MRI. So the first thing that we noticed was that if you treated these KP tumors with SHP-099, the SHP2 inhibitor, as a single agent, we were able to achieve disease control. The tumors really didn't shrink, but they didn't grow over this 2-week period, whereas the other tumors grew quite large and got to the point where the mice were in extremis and had to be sacrificed. Now, the SHP2 inhibitors, in addition to this effect on the tumor, also increased intratumor T cells. They decrease, sorry, I'm bad with this. So they decrease the bone marrow-derived, infiltrating bone marrow-derived macrophages that in general were polarized in an M2-like immunosuppressive manner. They decreased alveolar macrophages, which are also immunosuppressive. So decreasing the immunosuppression would be expected to be good from a therapeutic standpoint. And they increased intratumor B cells. I don't have time to show you, but in the paper, we depleted those B cells. And again, these B cells are exerting an anti-tumor effect. So these are all good effects of SHP2 inhibitor. But we also noticed that there was a substantial and significant increase in the GMDSC population, and GMDSPs are generally immunosuppressive cells. So we wanted to know whether any of these immune effects had any relevance to the disease. So what we did was we depleted either CD4 or CD8 cells, and we were able, but either of these depletions partially abrogated the inhibitory effects of SHP2 inhibitors, and they also substantially shortened the survival of these mice to SHP2 inhibitors alone. So we wanted to know what the T cell phenotype was in a little bit more detail. So what you can see is that the T cells that are in the TME aren't expressing any increase in standard classical exhaustion markers, but they actually were there was actually a relatively small population of effector T cells compared to naive T cells. And even the effector T cells that were there had very little cytolytic polarization, and the CD4 cells were largely not Th1 polarized. So these were not very potent T cells, which probably explains why they're exerting a relatively poor effect as anti-tumor agents. So, but as I mentioned, the SHP2 inhibitors also led to an increase in GMDSCs, which could be potently immunosuppressive. So we wanted to know what the effect would be of depleting these potentially immunosuppressive cells. So we first treated with a combination of, anti-LY6G and anti-RAD antibodies, and you can see that we successfully depleted the GMDSC population substantially. And when we do this, you can see that there's an increase in overall T cells, and these T cells are proliferating. And there's a polarization of these T cells now to CTLs as monitored by granzyme B and T-bet staining in the CD8 population. And there's also a substantial increase in Th1 polarization. So we wanted to know what the mechanism was for this increase in GMDSCs. So we purified the tumor cells from SHP-099 treated mice, and we did RNA-seq, and we noticed that two chemokines, CXCL1 and CXCL5, were substantially upregulated in the SHP-099 treated tumor cells. And these chemokines are known to be potentially trophic for GMDSCs. So in order to see whether, in fact, the action of SHP-099 in the tumor cells were having tumor cell non-autonomous effects to increase GMDSC immigration, we generated SHP-099 cells expressing the drug-resistant mutant, which we call TMQL. And you can see that this mutant is, in fact, drug-resistant because in contrast to the effects of SHP-099 in suppressing ERK activation in wild-type KP cells, when you express this drug-resistant mutant, there's no suppression. And consistent with the idea that the tumor cells were responding to SHP-099 expression by increasing CXCL1 and CXCL5, you can see that CXCL1 and CXCL5 induction is abrogated when you put in this drug-resistant mutant. And we then injected these cells into mice, and you can see that again, there's no effect on the increase in anti-tumor T cells. So this effect is not due to an effect in the tumor cells, it's an extrinsic effect. There's no effect on anti-tumor B cells. So this some combination of either the T cells or B cells or these other effects are happening outside of the effect on the tumor. The alveolar macrophages also go down, as do the bone marrow macrophages, but now the GM-DSC population is not induced in response to SHP-099. So these data suggest that SHP-099, by increasing CXCL1 and CXCL5 expression and production by tumor cells, causes an influx in GM-DSCs, which are potently immunosuppressive. So we wanted to know the mechanism by which SHP2 inhibition could lead to upregulation of CXCL1 and CXCL5. And we noticed that in the upstream regulatory sequences of CXCL1 and CXCL5, there were potential REL-A binding sites, and REL-A binding sites bind NF-kappa B. And so we hypothesized that NF-kappa B activation was the mechanism by which these chemokine genes were induced. And so we transfected KP cells with a reporter for NF-kappa B, treated them with SHP-099, and found that, in fact, there was a significant increase in NF-kappa B activity. And we wanted to know if this effect was mediated via a MEK-dependent pathway or MEK-independent action of SHP-099. And so we treated the same cells with trametinib, and you can see that there's equal induction of NF-kappa B when you treat with trametinib. So these data suggest that SHP-099, by inhibiting the RAS-ERK pathway, leads to an ERK-dependent increase, or at least a MEK-dependent increase in NF-kappa B activity. And in order to test whether NF-kappa B activity was in fact the mechanism, we inhibited IKK with the ML120B inhibitor, and you can see that if you block IKK, you inhibit the ability of SHP-099 or trametinib to induce CXCL1 and CXCL5. And we wanted to see if there was any direct evidence that SHP2 inhibition induces NF-kappa B activation. And so we looked at I-kappa B phosphorylation and P65 phosphorylation. And you can see that after a delay, which probably has to do with the induction of the production of a cytokine or growth factor that stimulates NF-kappa B, there's increased NF-kappa B signaling. So we wanted to know if this was just true in mice. So we tested a number of different human cell lines and human lung cancer cell lines, and just show you two on this slide. The GROW family of chemokines includes CXCL1 and CXCL5. And these chemokines all bind to the same receptor, CXCR1 or 2, the same chemokine receptors. And so what you can see in this slide is that treatment with SHP-099 in the sort of orange boxes, with trametinib in the blue boxes, or since these are G12C expressing cell lines, with the Mirati G12C inhibitor, MRTX849, increases the activity of these GROW family chemokines. So this is a general effect of RAS-ERK pathway inhibition. So in order to see whether this general effect of RAS-ERK pathway inhibition means that RAS pathway inhibition or even growth factor pathway inhibition will lead to increased GM-DSC activation and infiltration in the lung, we tested this in several different mouse models. And you can see that, as I showed you earlier, that treatment of KP cells with SHP2 inhibitors increases GM-DSCs. The same is true by interfering directly with MEK. So treatment of KP tumors with mice-bearing KP tumors, trametinib leads to increased GM-DSC infiltration. Likewise, treating EGF receptor mutant tumors with trametinib leads to increased GM-DSC activity infiltration. And a model that we generated earlier of KCP tumors, these are G12C tumors, treatment with the G12C inhibitor increases GM-DSC. So we think this is a class effect of inhibiting the RAS-ERK pathway in non-small cell lung cancer cells. So these data suggested that if we could block the effect of this chemokine induction, then we might improve the T cell response in these non-small cell lung cancers. So we were able to get a CXCR1/2 small molecule inhibitor called SX682 from a company called Syntrex. And what you can see is that when you treat mice with the combination of SHP-099 and the CXCR1/2 inhibitor, that you get a partial abrogation of GM-DSC infiltration. Now, those of you with good memories will notice that this is not as severe of a block of GM-DSC infiltration as the LY6G depletion, but it's nevertheless is a significant decrease in GM-DSCs. And this blockage is enough to cause a significant increase in T cell infiltration. The T cells start proliferating as they did with the GM-DSC depletion. There's an increase in effector T cells at the expense of naive T cells, and there's increased CTL function in the T cells and increased Th1 polarization. And this leads to substantially improved efficacy. So you now see significant shrinkage, like at RECIST levels for SHP-099 plus SX682 compared to either single agent alone. And this leads to a more than doubling of survival of these mice, but of course you'll notice that they're not cured. So we noticed, as I said earlier, that the GM-DSC effects were only partial of this SX682 inhibitor. So we wanted to know whether there was a specific subset of GM-DSCs that was being impaired by blocking the CXCR1/2 receptor. And so we did single cell RNA sequencing on tumors that were treated with either single agent or the combination. And what we noticed is that whereas SHP-099 caused an increase in many GM-DSC populations, only one population was abrogated by the co-treatment with SHP-099 plus SX682. And that's this so-called N4 subclass of GM-DSCs. And this particular subclass is probably the most potently immunosuppressive subclass because it's S100A high, which had been previously shown by the Gabrilowitz lab to mark a particularly immunosuppressive form of MDSCs. It also expresses CYBB, which is a subunit of NADPH oxidase, and in particular NOX2, which is also potently immunosuppressive, and also expresses lipocalin-2, which chelates iron in the tumor microenvironment and is also thought to be potently immunosuppressive. So we believe that this particular subset is blocked by CXCR1/2 inhibition and has salutary effects on activating the T cells. So what about the T cells? Are they also heterogeneous in the TME? So again, we looked at our single cell RNA sequencing data, and we identified two clusters, cluster 5 and cluster 2, that were increased upon CXCR1/2 inhibition. Cluster 5 is predominantly marked by cell proliferation markers. So these are proliferating T cells, but they're not they don't have a lot of effector markers. The other cluster, cluster 2, which you can see up here, is marked by the expression of granzyme B, and so it's presumably an effector cytolytic function. But we also noticed that it expressed two cell surface proteins, KLRG1 and KLRC1. KLRC1 is also known as NKG2A. So KLRG1 has recently been shown to mark a class to be on effector T cells, and that effector T cells that lose KLRG1 differentiate into memory T cells. So it's thought that KLRG1 may be a checkpoint receptor for preventing memory T cell differentiation. And NKG2A, although it's best known as an inhibitory receptor on NK cells, has recently been found to have a checkpoint inhibitor action on T cells as well. And this has been most vividly demonstrated by the recent results of a controlled phase 2 clinical trial, which showed that the anti-NKG2A blocking antibody monalizumab has increased efficacy in non-small cell lung cancer. So we believe that potentially combining NKG2A blockade with SX682 and or SHP2 inhibitor may be actually quite effective. And I should have mentioned this earlier, but the combination of SHP-009 and SX682 is quite effective in this model. Okay, so we wanted to know if any of this is true in patients as well. So we actually were very fortunate to be able to get access to some of the data from Mirati Pharmaceuticals. And what we noticed was that in 5 patients, 4 out of the 5 patients who had on-treatment biopsies showed an increase in CXCL1 and CXCL6. CXCL6 is the human analog of CXCL5. And very interestingly, the higher the level of CXCL1 and CXCL6 induction compared to the pre-treatment biopsy, the lower the response, or the lower the duration of response. And we also were able to infer the neutrophil number by using the algorithm MCP counter, and all of the treated patients showed a significant increase in the neutrophil or granulocyte, probably GMDSC population. We also were able to obtain single-cell RNA sequencing from a number of patients on study with the Amgen inhibitor at our institution, or treated with the Amgen inhibitor, I should say. And again, we noticed by single-cell RNA sequencing that CXCL1 and CXCL6 were induced in tumor cells primarily in patients on study, and in all of the 4 patients who were on the G12C inhibitor, they showed an increase in neutrophils, at least in inferred neutrophils from this single-cell RNA sequencing. So to summarize the first part of the talk, what we find when we treat lung tumors with a SHP2 inhibitor is that there's a decrease in alveolar macrophages, which are immunosuppressive. There's a decrease in M2-like bone marrow-derived macrophages, which are also immunosuppressive. So these are all good effects. And there's an increase in T cells and an increase in B cells. So all of these effects are potentially good for therapy, but there's also a concomitant increase in GMDSCs, which are potently immunosuppressive on the T cell population. And what we found mechanistically is that if you inhibit SHP2, KRAS, or MEK, or for that matter, EGF receptor, which I didn't have a chance to show you, that leads to an indirect increase in NF-kappa B, presumably because you block production of a cytokine or chemokine that's activating NF-kappa B. And this increase in NF-kappa B activity leads to an increase in CXCL1, CXCL5, or other Gro family chemokines. That leads to the production of the release of these chemokines, and where they act on CXCR2, on GMDSCs or progenitors of GMDSCs. And if we block with a CXCR2 inhibitor and an SHP2 inhibitor, this population is diminished, and that leads to an increase in Th1 polarized CD4 cells and an increase in CD8 T effector cells. These effector cells, however, express 2 potential inhibitory receptors, KLRG1 and NKG2A, and potentially interfering with one or both of these could improve this co-therapy even more. So again, in words, SHP2 inhibitor and CXCR1/2 inhibitor improve survival in KRAS and osimertinib resistant EGF receptor mutant non-small cell lung cancer. GEMS, I didn't have a chance to show you the EGF receptor mutants because I wanted to get to the second part of the talk, but that's also true. You can see it in the paper. And the osimertinib SHP2 SX682 triple combination evokes a stronger T cell response and delays recurrence upon drug withdrawal in OC-sensitive EGF receptor mutant GEMS. Similar effects are seen in non-small cell lung cancer patients treated with 2 different G12C inhibitors. Unfortunately, as I mentioned, these combinations are still not curative in mice even, and there's several possible reasons why. One is that there's inadequate neoantigens in these mouse models and or suboptimal immunogenic cell death. There probably would be more neoantigens in the human setting, so maybe this would be more efficacious in humans than in mice. But we believe that it's more likely that either the KLRG1 or NKG2A mediated checkpoints and or classical exhaustion need to be targeted as well to move from increased efficacy to potentially cure. Okay, so in the final part of the talk, I'd like to turn to a news story where we've posted the initial parts of this story online in BioRx, but we were asked to do another half a year's work, which we're submitting hopefully this weekend. Okay, so this has to do with these resistant cells that have come to be known as drug-tolerant persisters. And these are cells, so if you treat oncogene-bearing cells or in culture with targeted agents, almost all the cells will die, but a small population of cells survive in a non-proliferative state, And in some cases, if in the continued presence of the drug, they can grow out into this so-called drug-tolerant expanding persister state. And again, with continued culture, they can potentially acquire resistance due to secondary mutations. The drug-tolerant persisters and the classic DTEP state are both reversible and thought to be induced epigenetic changes. So we had several questions when Anderson Chang, the student who did this work in my Toronto lab, and it's now been continued by Jiayu Zhen, a postdoc in my New York lab, is first of all, do HER2 positive breast cancer cells give rise to DTPs and or DTEPs? If so, how did these cells originate? What are their properties and vulnerabilities? And do they exist in patient tumors? So we tested 10 different HER2 positive breast cancer cell lines, and we found that half of them formed DTPs. We call these type 3 lines. Only one of these type 3 lines over the 8-week period that we tested this formed drug tolerant expanding persisters. So most of what I'm going to talk about, or essentially everything I'm going to talk about today, has to do with this DTP state, not the DTEP state. So we first did RNA-seq, bulk RNA-seq on the DTPs compared with the DTPs, and we found that they activated or appeared to activate 2 distinct transcriptional programs. One set, which stemmed from the HER2 positive ER- cell lines in this collection, had a predominantly EMT-like transcriptome. And the genes that were induced by this EMT-like transcriptome were enriched for SMAD4 and SOX2 binding sites. The other set of lines had a predominantly luminal-like phenotype, and these genes were marked by hallmark estrogen response, or these genes were marked by estrogen receptor binding sites. So now, while this work was in revision, there were 2 papers that were published, including one from Konstantin Mitsiadis at your institution, actually, that identified a diapause-like DTP state in cells that are able to survive chemotherapy and some other therapies. And then Ari Melnick identified a senescence-like state in AML cells. And these groups all published signatures, and we wanted to know if there was a relationship between these signatures and the luminal and mesenchymal signature that we saw. So what I have found was that hallmark MYC targets, which were also identified as downregulated in all of these studies, were in fact downregulated and were identified in unsupervised analyses of our HER2 TKI DTPs. But none of the other signatures came out of unsupervised analyses, although supervised analyses did reveal an enrichment for the diapause signatures in both the mesenchymal-like and the luminal-like DTPs. So we think that the diapause signatures occur here too, but the dominant phenotype are these luminal and EMT-like transcriptomes. So we wanted to know, as had been done in the O'Brien and Mitsiadis papers, whether the DTPs were arising stochastically or whether they were the consequence of some pre-existing hierarchy. So we did barcoding experiments, and the concept here is that if there's a hierarchical organization, then when you label cells with barcodes, only a small fraction of the barcode should be recovered at the end of the experiment. But if it's stochastic, basically almost all the barcodes should be recovered. And you can see clearly just by inspection that almost all the barcodes arise stochastically. So one of the good things about BioRx is that people actually read them occasionally. And we were contacted after we posted our original preprint, which only had the barcoding data on BT474 cells, by Abhi Singh, who's a computational biologist at the University of Delaware. And he said he had taken our data and analyzed it, and that he said he totally agreed with the conclusions that I'm going to tell you in a second, that we had derived from single cell RNA-seq, that there was actually a sort of primed or pre-DTP state in these cultures, and that he could even analyze more of these if we gave him more data. So basically, I don't have time to go through his analysis today, but using Luria-Delbruck effectuation analysis, he's able to conclude that although in fact DTPs arise stochastically, that there's actually a prime state consistent with our single cell RNA-seq data that I'll show you now, and that this prime state has a heritability of between 2 to 4 generations, or 2 to 6 generations, depending on the cell line. So that's just an interesting esoterica to keep in mind for the rest of the talk. Okay, so as I mentioned, we did single cell RNA sequencing to see if there was any hierarchy in this in the untreated and treated cells, and to see how DTPs became DTPs. So what we noticed was that if you considered the DTP up, if we compared the DTP up DGs, which are the genes, the differentially expressed genes that are upregulated in DTPs compared to parental cells, as you treat with lapatinib, the TKI inhibitor that we used for the initial studies, there was a progressive increase in the expression of the DTP up DGs and a progressive decrease in the DTP down DGs. But when we looked closely at the untreated BT474 cells, we noted that there was a bimodal distribution, or there appeared to be a bimodal distribution, in that some cells already had substantial downregulation of the DTP down DEGs. Then we also noticed by unsupervised clustering that one cluster, cluster B, had an increase in level of expression in the DTP up genes and a decreased level of expression in the DTP down genes. So in other words, there were cells that showed transcriptional similarity to the DTPs prior to lapatinib treatment or prior to HER2 TKI treatment. So we interrogated this pre-DTP cluster, and we found that it was highly enriched for G0 signature genes. And this is probably most vividly illustrated here, which is if you compare the what we call the BT474 up and down scores, which are a score for the upregulated genes and the downregulated genes, and then the combined score is just subtracting the down score from the up score to give you a combined score. You can see that there's enrichment in these G0 cells for pre-expression of the DTP signature, suggesting that there is a pre-existing state that somewhat resembles the ultimate DTP state. And we have a similar observation in the other type 3 HER2 positive breast cancer lines, which you'll be able to read online hopefully next week. So we then looked at pseudotime trajectory analysis. Well, first we wanted to know if we could find a marker, a cell surface marker, to more directly interrogate whether there was in fact a pre-DTP state. So we compared the transcriptomes of the pre-DTP cluster B with the BT474 DTPs, and we identified overlapping genes, which included 2 cell surface markers that I'll talk about, actually more than 2 cell surface markers, but in particular MPY1R, which is shown in red, and then the other cell surface markers are shown in bold. And I'll just mention that everything I'll tell you about MPY1R holds for ABCC5, which we also did flow cytometry expression on, FACS purification on. So the first thing that we noticed was that if you looked at trajectory analysis of the single cell RNA sequencing, the DTPs, most of the DTPs came from this G0 population and gave rise to the so-called DTP1 state. Now there's also a very small, about 10% of the BT474 cells gave rise to this DTP2 state, which actually is enriched for G2M genes. These may represent a small number of so-called cycling DTPs that were recently described by the Regev and Brugge lab, and also similar cells were described by Sabrina Spencer's lab in melanoma. Although we don't see any expansion of this population over 8 weeks, as I said earlier, so we can't be sure that these are not just permanently arrested cells or cells on the way to die. But interestingly, if you look at the MPY1R high cells, they also are enriched for giving rise towards the DTP state. So in order to test directly whether there were pre-DTPs, we used flow FACS sorting to sort out low populations, high populations, and mid populations of MPY1R expression. And what you can see is that there's a substantial enrichment for the ability to form DTPs in the MPY1R population. And these populations do, over a 14-day period, regenerate the respective surface expression of MPY1R, consistent with the idea that they have a stochastic organization. So we looked at what happens to the MPY1R cells, basally and over time, in response to lapatinib. So the first thing is you can see that there's a substantial enrichment for DTP genes in the MP1R high untreated cell population. And these cells are also enriched in the untreated population for estrogen response early genes. And if you then treat these cells with lapatinib, you can see that over time, in an ER-dependent fashion, because it's blocked by fulvestrant co-treatment, there's a further induction of MPY1R. And these MPY1R high cells are enriched for G0, G1 cells, consistent with the analysis that I showed you earlier of single-cell RNA-seq. So interestingly, we looked at these other signatures, the MYC target genes, the diapause genes, and the Melnick chemotherapy-sensitive genes. And again, we looked in all of these are all untreated cell lines. And you can see that there is, again, downregulation for the MYC target genes in all of the cell lines, untreated. And there's an increase in the diapause score in all of the cell lines. And all of these are significant. And these correspond to the G0 population. And there's also an increase in the Melnick CISG score. So these data suggest that there are cells that have the properties of DTPs, or at least parts of the properties of DTPs, before you treat the cells with TKI. So we wanted to know if this was actually also true in patients. And so we were actually lucky that our colleague Sylvia Adams, in collaboration with Kwok, had actually done some single-cell RNA sequencing on a couple of HER2-positive patients. And again, we found that the the cells that were annotated for the G0 signature had a pre-enrichment for mesenchymal DTPs. These are HER2-positive, ER-negative patients, and a decrease in the DTP down genes in the G0 population. And this was true in both of the patients we could look at. And they were then very fortunate to get access to the bulk RNA sequencing data from the NEO-ALTO trial. The NEO-ALTO trial is a neoadjuvant trial that had a lapatinib alone arm. And what you can see is that patients who have a high G0 signature score pre-treatment have a lower chance of pathological complete response, consistent with the idea that they may have increased ability to generate a drug-tolerant population upon lapatinib exposure. So how do we deal with these DTPs? Well, I already told you that when you pre-treat with fulvestrant, that you block the ability of MPY1R cell and the MPY1R high cells to become even higher. Not surprisingly, if we co-treat with lapatinib plus fulvestrant, you dramatically decrease the ability to generate DTPs in the HER2-positive, ER-positive cell lines. So this is basically rediscovering what clinicians already know, because as you probably know, one generally treats ER-positive, HER2-positive patients with a combination of anti-HER2 and estrogen-responsive agents. So there's nothing really new here, except that this provides sort of a mechanistic underpinning for why that might be the case. The other thing that we found, which I don't have time to go through in detail, but I'll just show up here in this diagram, is that we also found that the PI3 kinase pathway was considerably rewired in the BT474 DTPs. So instead of AKT being the predominant activator of the mTOR pathway, it turns out that SGK3, which is an ER-responsive gene, is upregulated substantially, and it's now the major kinase that inhibits TSC2 and leads to mTOR activation. And so if you look on the right-hand part of the slide, you can see that if you co-treat DTPs with PI3 kinase inhibitor and lapatinib, you get pretty good effects by themselves, but you don't get much of an effect with a MEK inhibitor, with a AKT inhibitor. By contrast, if you treat with a SGK3 inhibitor, then you do get a good effect. And interestingly, it turns out that the mTOR pathway is also rewired in the mesenchymal cells, but it's not through SGK3, and we still don't know what the kinase is that does this. So to summarize the second part of the talk, we believe that DTPs are not simply drug-induced, but there's actually a prime state, which we call pre-DTPs, characterized by partial activation of ER target genes in the case of the luminal-like DTPs, and diapause and senescence programs, and characterized by a G0-like expression signature. Now, we don't have firm proof that this is equivalent to the G0 population that exists in cell cultures as defined by Sabrina Spencer and Toss Meyer, or whether it's just a different population that has a G0-like signature. But we know that it does have a G0-like signature and partial expression of these genes. Then upon exposure to HER2 TKIs, there's further induction of ER target gene expression and a rewired PI3 kinase mTOR pathway to promote survival. And these cells can sit around and presumably on drug holidays re-expand or possibly acquire additional mutations and lead to recurrence. Now, I want to point out that very recently, like I think 2 weeks ago, the Brugge and Regev labs, did a completely separate study focused on cycling DTPs. They also identified heterogeneity in the untreated cell population because these cycling DTPs appeared to come from a separate population in the original starting population. Now, I should mention that these cycling DTPs probably give rise to what we call may give rise to what we call DTPs. And I would say that they actually studied 2 of the cell lines that we studied in our study and found that they did not give rise to cycling DTPs at all. So it's the 2 cell lines that didn't have any cycling DTPs consistent with our finding and focus on non-cycling DTPs. So to summarize the second part of the talk, HER2 positive cell lines form 2 types of drug tolerant persisters in response to HER2 TKIs, luminal-like and mesenchymal-like. DTP generation is stochastic, but most DTPs emanate from pre-DTPs or primed DTPs characterized by a G0-like signature and partial expression of a DTP program with limited heritability for 2 to 7 generations, depending on the cell line. The luminal DTP program is further induced upon exposure of these prime cells to drugs. So we would favor a predetermination or priming induction model of DTP generation. DTPs rewire their signaling pathways from receptor tyrosine kinases to mTORC1 to enable survival. And consequently, DTPs and pre-DTPs presumably have unique vulnerabilities compared with bulk parental cells. And so obviously the major question is what's the molecular, presumably epigenetic basis for the priming of the pre-DTP state? And that's what we're actively working on with our new collaborator, Jane Scott, right now. So I just want to summarize again, acknowledging the people who've done the work. So the ovarian cancer system that I alluded to earlier was developed by Shuang Zhang. She's now junior faculty at Guangzhou Medical University, and she was assisted by a large number of collaborators listed on this slide. I want to note that Sonia Iyer, when she was a postdoc in the Weinberg lab, also had a similar study, and Shuang and Sonia and Bob and I collaborated on this study extensively. And I want to acknowledge Shenjing Zhu, my former postdoc who's with Miles and Shirley, for helping us on some of the bioinformatic analysis in the human population in that study. And the work that I talked about on RAS signaling was a collaboration between Quan Ho Tang and Shuai Li in the Wang lab. And again, these are our collaborators. I want to acknowledge in particular John Zabala and Dean Maeda at Syntrex for giving us SX682, and Jamie and Peter for sharing their data from the Mirati inhibitor trials. And finally, the last part of the talk was started by Anderson Chang when he was a grad student of mine in Toronto, and he now works as a scientist at AbbVie in San Francisco. And the work's been taken on by Jiayu Zhen, who just became a new mother and is home helping to write the paper while she's breastfeeding her baby. And these are the collaborators. I want to particularly acknowledge Benjamin Habe-Keynes and Arvin Mayer and Chris and David, who helped us with the Neo-Alto data, and Abby Singh, who did the Luria-Delbruck fluctuation analysis. And thank you for inviting me, and I'll take questions from the chat.
Great. Thank you so much, Ben. This is outstanding. So I wish like Zoom has a clapping, applause this the feature. But
can I answer Dan Tennant's question?
Yeah. So this. Yeah. Okay. Yeah.
Dan wants to know if I can get him a job. Dan, that is Dan. That is very much Dan. Dan, I thought you had, I don't know how many more jobs you can handle. I thought you had 2. Okay.
Okay. It's offline. You guys can talk.
Yeah, he's going to ask.
Okay.
He's going to ask me if I can get him a raise next.
But Dan is here. I thought Dan's Singapore. Okay.
Who knows? He's interwoven.
Okay. Okay, so second question. We have a few minutes, you know, people can keep sending questions if we cannot, you know, so no time, and then Claudia will forward your questions to Ben email. Okay, so the second question, Danish, what other, you know, phosphatase other than SHP2? Where is that? Okay.
Yeah.
Do you see have this relevance in cancer therapy, immunotherapy?
Yeah. So obviously, so probably the best documented one is PTPN2 or T cell phosphatase, which has both cell autonomous effects on the tumor for increasing tumor immunogenicity, and also has salutary effects on T cells. So not acting on by basically being a negative regulator of cytokine signaling. So loss of negative regulation of cytokine signaling can be pro-immunogenic. Also, well, I don't know if that's published yet, so I shouldn't say what the other one is. So those, and probably PTPN22 has recently been suggested to be a immunotherapy target by, I'm blocking on her name, from Hopkins, pancreas cancer. I can't believe I'm blocking on her name, but I'm getting old, so I can't remember anything anymore. So.
Okay, next question from Stengu. SHP2 was also reported to be involved in CD28 and PD-1 signaling. Did you examine the T cell autonomous effects of SHP2 inhibition? This is question one. And then he has a second question. Did you observe elevated antigen presentation?
So those are good questions. So first of all, I just want to say that so the role of SHP2 so initially it was thought that SHP2 was the effector of PD-1. So SHP2 definitely binds to the PD-1 ITIM residues. However, SHP2 T cell specific knockout mice are still perfectly capable of responding to PD-1. And that was published by 2 groups already. And, you know, we find the same thing in our T cell knockout mice. In fact, if anything, the SHP2 knockouts may be more responsive to PD-1 in anti-tumor models. So it's not essential for PD-1 action. In terms of whether we examine the T cell autonomous effects of SHP2 inhibition, what we're doing is we generated an inducible knock-in of a drug-resistant mutant of SHP2, so we can basically deconvolve the effects in all parts of the tumor microenvironment. So we knock in the drug-resistant mutant under the control of a lock-stop box cassette. So that will be that will be my retirement project. Okay, so the second question is, we have not looked at that, but that's a
good one. Yeah, that's interesting. Okay, this is also actually my question, but okay. Next is Tom Rook. Tom Rook, great talk, Ben. Do the DTEPs expanders grow out due to the selection of rare cells? Yeah. With pre-existing resistance, epigenetic changes. Yeah.
In our system, we only found one. We only saw one cell line give rise to DTPs over the eight-week period. So we don't really have any, we didn't really study them in much detail. We just looked at the DTP state. That being said, Jeff Settleman, when he was at MGH, you know, he pretty clearly showed that at least the PC9 line, the non-small cell lung cancer line, that was the original line used by, I should have given Jeff Settleman credit, that paper was appreciated when it first came out on DTP. Somehow his name disappeared from that slide. I should add it back. But Jeff Engelman followed up on the original Settleman paper, and he found that PC9 cells, which are the lung cancer line that a lot of people have used as the model for DTPs, that stocks of those cells had a small population of already drug-resistant cells, and that they gave rise to, they did a large, you know, clonal analysis, and they found that early expanders, early resistant cells came from that drug-resistant population that was maintained in the PC9 stocks. But then there was a late DT piece, and they didn't come out until, like, I forgot, much longer than 8 weeks, like 12 weeks. So it's possible that if we waited that long, that the other 4 lines that did not form DTPs would form DTPs. I also want to say one other thing about experimental details. So, some of the studies that were done with lapatinib resistance early on did not use the peak serum levels of lapatinib. Okay, so we endeavored to be very careful and match the peak serum levels of lapatinib to generate DTPs. So, you can get resistance, or, you can get adaptive resistance to it. You don't give enough lapatinib and it'll grow out. So it's really important to pay attention to little things like that.
Okay. So Ben, if this is okay with you, there are 2 questions. Let's just do it.
I'll do it quick. So
okay, yeah.
My former graduate student cannot ask me questions. Okay.
Okay. So you want to do Joan's question?
Okay, so I'll answer Joan's question. No, I'll answer the other one. So we don't know that.
Okay.
So we don't know that. That's obviously very interesting.
Mm-hmm.
So Joan's question is, can you explain again how the reanalysis of your experiments concluded stochastic generation of persisters identify prime cells? So Joan, I probably went through that a little bit too quickly. So what the Luria-Delbruck fluctuation analysis revealed was that there were populations of cells that had a heritability of between 3 to 6 generations that behave differently than the bulk population. And that plus the single cell RNA-seq data leads us to infer that those are the cells that are primed. But the single cell RNA-seq data and the direct sorting with 2 different cell surface markers really, I think, are the conclusive evidence that there are prime cells.
Great. Thank you so much. So let's thank you again, Ben, for the outstanding talk. And then today, actually, we had a lot of more than 100 turnover now. Okay, great. Have a nice evening. Thank you.
Bye.
Okay, bye.